welcome to part 3 of the second module for this unit. Here we're going to talk over just a few things, really what happens after the process of meiosis in particular. So in part 2 we talked about mitosis and meiosis, where mitosis is a type of cell division where you take somatic cells and you make more somatic cells from it. But meiosis is a process where you take sort of somatic-like cells and you end up making gametes from it, or your sex cells, so that being the egg and the sperm. Now that the egg and sperm have been made, what happens with them? And so if the egg and sperm sort of end up being used as sort of they're intending to be used and they join together, that's a process called fertilization. Now, once an egg has been fertilized by a sperm, it's going to form a cell. It's a somatic cell that is, so it's got 46 overall chromosomes, and it's specifically called a zygote. All right, and this is immediately after you've got your egg. It's been fertilized by the sperm. It is now a zygote. It is a diploid cell, which means it has 2 of each type of chromosome. So that's 23 chromosomes from mum in the egg and 23 chromosomes from dad in the sperm, and that gives a total of 46 chromosomes. Now, this is essentially now a somatic cell, and so it wants to make more somatic cells, so it's going to go through mitosis. And so once we've got the zygote being formed, it actually quite rapidly starts to divide through mitosis, forming an embryo and eventually a baby. There are certain names used at certain stages of development, so fetus, embryo don't always get used at the same time, and technically baby, I believe, isn't meant to be used until once it has been born, but it is a baby. So our zygote's rapidly dividing through mitosis to make a new little person. And it's really important here that those 2 gametes only have those 23 chromosomes. And I mentioned this in one of the other videos. If it doesn't, and let's say they were each like a somatic cell and they had 46 each, you would end up with a zygote that has 92 chromosomes. And that's not going to work. Okay, you can't have a little mutant baby like that. It won't develop in the uterus. It will essentially just not progress and the body will do away with it. So you have to have gametes that only have 23 chromosomes each. So when they combine and form the zygote, they end up with a total of 46 chromosomes that can be grouped into 23 pairs, and then that will develop into the baby. So speaking of babies, most pregnancies, you will end up with it's a single egg that is fertilized by a single sperm. And so you end up with one child being born. But occasionally we have multiple births, which the most common type of multiple birth being twins. Now, we'll talk about twins, but you can sort of extrapolate this a little bit for triplets or quadruplets and so further. But twins generally fall into 2 categories, identical or non-identical. And they fall into these categories because one of 2 things can happen. Now, if we're talking identical twins are genetically identical. They're essentially like little clones of each other. And what has happened here is this first image down the bottom. It is 1 egg and 1 sperm, and it forms 1 zygote. But at some point along the process of development and everything, that zygote, as it's dividing, gets a little bit confused. And instead of just continuing division and making 1 baby, it sort of resets at one point and suddenly thinks it's actually 2 zygotes, and they each develop independently of each other, and so it ends up forming 2 embryos, and we end up with 2 genetically identical babies because they were formed from the same egg and the same sperm. So that's identical twins. On the other hand, we also have non-identical twins, and so non-identical twins are twins where they don't necessarily the exact same set of genetic information. If you have twins where one's a boy and one's a girl, they are non-identical twins. Hopefully that's fairly obvious, but just in case it wasn't. All right, so with non-identical twins, you actually have 2 egg and 2 sperm. So you have 2 eggs that are fertilized by 2 different sperm. And so those 2 eggs that have been fertilized form 2 different zygotes. And they end up developing at the same time in the same womb, but they form 2 separate babies. And so that's what this second image is showing. And so the babies here are genetically different. Now, non-identical twins can be the same gender. It's just if you have twins of different genders, they're obviously non-identical. If you had 2 boys or 2 girls, they could either be identical or they could be non-identical. You would need to look at the genetic makeup to work out which they are. Sometimes you may not need to go that far because it's quite obvious, because they look identical, or they don't look identical. Other times, if you're not sure, because of other factors, you would have to go look at their genetic information to work it out. So the next thing we'll talk about with, say, a zygote or a baby is how is the gender or the sex of that baby actually determined, or has it determined in people? And I did mention this earlier, but I'll go back over it here. It's actually fairly simple. There are sex chromosomes in our somatic cells that are responsible for determining our sex or our gender. Now, if you're female, your sex chromosomes are X and X. You have 2 copies of an X. That's all you've got. And what that means is your eggs will only ever have an X as a sex chromosome in them. It can't ever have a Y. Not possible. Girls do not have Y chromosomes. Not a thing. Males, on the other hand, are XY. And what this means is that their sperm could either have an X or it could have a Y in it. And so what that means is the father actually determines the sex of the child. So here you can see mum's made 2 eggs and both of them have X's. She can't do anything about that. So while mum's producing egg cells with only X's in them, dad, on the other hand, is producing sperm, and he's either going to put his X into the sperm or his Y chromosome is going to go into it. Now, if he fertilizes an egg with the sperm that has the X in it, he's going to produce a female child, so he'll have a daughter. If, however, he fertilizes the egg with a sperm that has a Y in it, he will produce a male child, so he'll have a son. So this is how sex is determined in human offspring. Most animals are the same. If we start talking about like birds or other organisms, there are some other ways that sex can be determined, but we won't dig into this now. Although if you've got some time in your hands, I do recommend looking at how sex is determined in clownfish. And then if you've watched Finding Nemo, think about that in a little bit more detail, because it's quite interesting and may ruin Finding Nemo for you. I apologize in advance if you go and look it up. All right, but as I mentioned in the other video, also this is what we're talking about here. This is the reason why Henry VIII was actually at fault for, never having male offspring, or only ever producing 1 male offspring, I should say. It wasn't his wife's fault, it was actually him not being able to produce a male heir. So sometimes we might want to look at a baby's chromosomes for a range of reasons. Maybe we want to look at a baby or an individual's chromosomes to see genetically what their sex is, or we might want to see if they've got any chromosome abnormalities or disorders and that sort of thing. Sometimes parents might want to do this before they give birth to a baby to see if the child's going to have any conditions they need to know about. The way we actually go about getting a copy of the chromosomes and presenting this image so we can analyze it is something called a karyotype. So a karyotype is a picture, basically, of all the chromosomes in a cell. Now, this is just a sort of drawing. It's not a real karyotype, but it gives you an idea of sort of how we might lay them out. Now, to take the image we need to do a karyotype, we may have to be able to see the chromosomes, and we need to do it before the cell's actually divided. So there's a very sort of small window where we can actually do this. So it has to be taken right before division, when the chromosomes are condensed and visible. So after DNA has replicated, but before mitosis has really started. And so we can take an image, take a photo using a microscope of the chromosomes in the cell, and zoom in close enough and take that photo. And then that can be taken, and the chromosomes can be sort of picked out one by one. They can be paired up, and they can be arranged in their pairs from big to small to form a karyotype. So in case you're wondering, how do we know what chromosome 1 is? Well, chromosome 1 is always going to be your largest chromosomes. And then it counts upwards, but as it counts upwards, the chromosomes actually get smaller and smaller. And so we can use these to help show gender and some genetic disorders as well. There's a couple of other things at play when we're trying to arrange the chromosomes, and particularly trying to pair them up. We look at the size, we look at the stripes on them. As you'll see on the next slide, they actually have little stripes. They look a little bit like barcodes. And also this position of the sort of middle pinched in bit called the centromere. Those are the 3 things we're interested in when we look at the chromosomes and want to pair them up. And that's the thing. So here is an image of an actual karyotype, and you can see our chromosomes, unfortunately, don't always play nice, and they don't always lie in nice sort of straight lines. You might see karyotypes where they look more like letters I, which is kind of what they look like here. They might look more like an X. It doesn't really matter. Don't worry about it too much. It will just depend on when the image has been taken and what's happening. Now, here you can see the stripes on them. They're the banding patterns, and you can sort of see on some of them more clearly than others where they're sort of pinched inwards at that centromere. They've been paired up for you, so you've got 1 all the way down to 22, and then you have the sex chromosomes over here. So looking at this karyotype, see if you can identify the sex the individual is, and then explain also how you know that, and also have a little look a little more closely and see if there's anything unusual about this karyotype, or that seems a bit out of place. And I'll come back and I'll talk you through it in a moment. So pause the video now and go and analyze this karyotype. Okay, so hopefully you've gone through and you've been able to answer those 2 questions. So let's talk about the sex of the individual this karyotype has come from. So I mentioned before that here we have our sex chromosomes, and you can see one is labelled X and one is labelled Y. Now, because that means we have an X chromosome and a Y chromosome, and we know only males have an X and a Y chromosome. So that is going to tell us that this is a male. So this karyotype comes from male, and we know that because it has an X and a Y, and only males have X and Y. Now, if you looked closely, and maybe if you counted up the number of chromosomes here, you would have found that this karyotype actually has 47 chromosomes, which is wrong. It should have 46. And what has happened here, number 21, instead of having a pair, has a triplet. It has 3 chromosomes, and that's not normal. If you look into it, you will find that having 3 chromosome number 21s is actually Down syndrome. So this is actually a karyotype of a male with Down syndrome. So this is that's one way that karyotypes can actually be really useful to help identify those sorts of conditions. Particularly if this was before the baby had been born, the parents could know beforehand that they were going to have a son with Down syndrome, and then that could help them prepare accordingly for that. So that's a really handy way we can use that. Alright, so we're now at the end of this screencast, and in fact all the screencasts for this module, and I have here a self-check quiz. So depending on how you're doing this, you may just answer this on a piece of paper, or you may have a quiz to complete, but go through and answer these questions, and there will be a video going through each of the answers once you have done that. If you've got a quiz, it will be released at the end of the quiz. So go through and check your knowledge from the past 3 videos that we've had in this module here. Alright, that is all from me from this, and I will see you in our next screencast. Bye!